this part of the lecture is going to be focusing on different types of cytoskeletal filaments that we can find within the cell. And there's three different types that we're going to talk about. But let's just start by kind of figuring out first, what is the cytoskeleton? Well, like it kind of sounds, or the name sounds, it's kind of a network of fibers that extend throughout the cytoplasm and often give support to the cell. So it says here they give mechanical support to the cell, and they can also help the cell with different types of motility, which utilize different types of motor proteins. And like I mentioned, there are three main types of fibers that can make up our cytoskeleton. Okay, so there's three main types of fibers that help to give the cell that strength, or that kind of fibrinous network that allow it to withstand the elements. And the three main types of fibers are microtubules, and we'll talk about those throughout the rest of the course. We have a type of cytoskeletal filament known as intermediate filaments. And then finally, we have these microfilaments. And this page is just, it's just there for your information and to get some visual kind of representations. But on the left-hand column, you have microtubules, and you can kind of see on the bottom how each of these differ in shape and what their actual images look like. In the middle, you have these microfilaments, and on the right-hand column, you have the intermediate filaments. So let's start with microtubules. What are microtubules? Well, again, they help to give shape to the cell. And microtubules are really important because they're going to help guide movement of different types of organelles. Now, the reason we're going to talk about these later in the course are that these are really important in cell division and in DNA replication. So it says in this last bullet, it says microtubules help separate the chromosome copies in dividing cells. Well, when we talk about cell division or DNA replication, we're going to talk about the fact that when we create new DNA, we have to move it to either side of the cell so that each new cell that's created has a copy of the DNA. And the way it's able to move is because of these microtubules. So just keep that in the back of your mind and understand that microtubules are really important in different types of movement, but they're especially important in helping chromosomes to divide. Now, intermediate filaments help, again, with cell shape, but these are important because they really help to kind of fix organelles in their place. So at the beginning of this lecture, we talked about a eukaryotic cell and how it has all these different parts that are kind of fixed in certain locations around the cell, and it's very organized. Well, they're able to stay there, and they're able to maintain that organization because of these intermediate filaments. Now, microfilaments, or they sometimes call them actin filaments, are built from different types of protein molecules. And it says here they're found in microvilli. Well, I think kind of the first thing I think of when I think of microvilli are kind of little fluctuating kind of cilia that you might find maybe in an intestine. So you want to have this movement within your intestines to keep food processing and to get food flowing. And that occurs because of these microfilaments. And it says microfilaments that function in cellular motility contain the protein myosin in addition to actin. So really important types of muscle proteins and all those different structures that assist us with movement come from these specific microfilaments. And this picture is just kind of interesting, but you can see again these microvilli at the very top of the picture. And those are just they almost look like little hairs, but they're little projections that help to move different substances throughout your body. And again, here's those myosin proteins that we find within microfilaments. And again, this is really important in different types of muscle contraction and just movement overall. So this is just a kind of an example of why these are indeed important. Now, if we're talking about the cell, we also have to mention these things called centrosomes and centrioles. And these are, again, going to be really important when we talk about cell division and DNA replication. It says the centrosome is considered to be a microtubule organizing center. Well, let's look over on the right-hand side of the page, and we see this kind of highlighted yellow region with these two cylindrical objects in it. Well, those cylindrical objects are those centrioles. And if you look closely, you can see that it looks like these centrioles are just kind of a bunch of straws that are glued together. That's what it looks like to me. But really, those little straws are supposed to be the microtubules. But you can see that they're kind of organized in almost like a star-like fashion. And so that's what it means when it says that a centrosome is a microtubule organizing center. It just kind of organizes those cytoskeletal filaments. And the centrioles together are what make up this centrosome. Now, cilia and flagella are basically specialized arrangements of microtubules and allow certain types of cells to move. And the first thing I think of when I hear cilia and flagella is I think of an egg cell and a sperm cell. In an egg cell, you have all these little projections that are coming off of it, and it helps the egg to move, through the fallopian tube and into the uterus where it can be fertilized. Well, flagella, on the other hand, is what you think of when you think of a sperm cell. You have this one kind of big tail, if you will, that helps that cell or that sperm cell to move through the body. So here's a much better picture than I can just describe without actually showing you something. But on top you have flagella, so you get this kind of one-directional movement of a sperm cell. And on the bottom you have this cilia, and you can see again they look like little projections coming off of the cell membrane, but they move back and forth and help that egg cell to relocate to a spot where it can become fertilized. Now, cilia and flagella share kind of a common ultrastructure, and that's the fact that you have this kind of microtubule formation, if you will. So if you look closely, on the left-hand side it says you have these microtubules that kind of make up the structure, and on the bottom picture you can see this kind of cross-section, and you see how you get these kind of triplets that exist of the microtubules. And if you look on the upper right hand picture, kind of the cartoon picture, you can see again kind of that it's not a triplet here, but it's a doublet formation that exists. So cilia and flagella can share a common ultrastructure in that they're composed of these microtubules. Really, that's what I want you to grasp is the understanding that cilia and flagella aid in cellular motility, but they're also kind of structured similarly in that they are composed of different types of microtubules.